Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasist from Department of Biotechnology and Bioinformatics, JP University of Information Technologies, Himachal Pradesh. Today I am going to talk about a module that's cell adhesion and communication. This module is placed in paper Thermodynamics and Living System. Adhesion is the process by which different substances interact and bind to each other. This property of binding also happens in biological cells, by which they attach to nearby similar cells and make an architect of tissue organization. A number of cell surface proteins like cell adhesion molecules mediate such homophilic, which means like binds like adhesions between cells of single type and heterophilic adhesions between cells of different types. Most cell adhesion molecules are uniformly distributed along the regions of plasma membrane. They contact other cells, and the cytosol facing domains of these proteins are usually connected to the elements of cytoskeleton. Present module provides information using the following objectives for such adhesion molecules and their presence in specific junctions of a cell. So, for that matter, following objectives were prepared to achieve these informations. Number 1, to understand the basic process of cellular adhesion and its related consequences. Number 2, to classify the cell adhesion molecules and elucidate their structure and functions. And lastly, the number 3, to elucidate the functions of different cellular junctions and their relative functions. Now, if you see that interaction of cells is fundamental in multicellular organisms that hold cells together. Molecular interactions at cellular level are essential for assembly of different cells and tissues. Cells either adhere together cell junctions, or they secrete some extracellular molecules that help them to bind together. The mechanism of cell adhesion is important as it governs cell shape, their arrangements and their strength. Cell junctions allow cells to exchange their biomolecules and communicate in order to synchronize their behavior and regulate gene expression. If you see that in connective tissues where the cell adhesion is rare and collagen rich extracellular matrix is abundant, that bears mechanical stress and allow cells to pull on it. Whereas in epithelial tissues, extracellular matrix is scarce and present in as basal lamina under the sheet of cohered cells. In these tissues, cells adhere together by means of cell-cell adhesion, and cytoskeletal filaments are present inside the cells which transmit signals inside the cells through adhesion sites. This figure shows the arrangement of epithelial and connective tissue. Figure A represents the basic lamina under the sheet of cohered epithelial cells and extracellular rich matrix connective tissues, while B is showing cell surface adhesion molecules which are interacting with cytoskeleton and extracellular ligands. Physical attachment between all types of cells is essential, but junctions are also required for diverse functions apart from just transmitting physical forces. There are 4 main types of cellular junctions which are based on their functions. Number 1, anchoring junctions, which may be of cell to cell adhesion and cell to matrix adhesions. Number 2, occluding junctions, which are required for sealing cells together. Number 3, channels, which forms the junction of connecting cytoplasm to adjacent cells. And number 4, cell signals relying junctions, which are required for cell communications. In today's module, we will focus on 3 junctions, and the last one, junction, cell to cell communication junctions, were discussed separately in another module. So, this figure shows different types of junctions according to their locations and functions. In these junctions, the above 2 junctions are primarily focuses on cell adhesions. Now, apart from cytoskeleton, proteins, each of above mentioned junctions require 2 types of proteins. Number 1, intracellular anchor protein that acts as an adapter between the cytoskeleton and the extracellular protein. And number 2, the transmembrane adhesion proteins, which are generally known as cell adhesion molecules or CAMs. These cell adhesion proteins have 3 domains. Number 1, intracellular domain, which is required for interaction with intracellular anchoring protein. Number 2, transmembrane domain, which is required for anchoring the proteins to the membrane. And number 3, extracellular domain for interaction with other CAMs or cell adhesion molecules. These molecules are involved in binding of cells or with extracellular matrix. These molecules are distributed along with plasma membrane and are required for cell binding at cell junctions. Binding between similar cell adhesion molecules on cell surface is homophilic binding, and between different cell adhesion molecule classes is known as heterophilic binding. There are different classes of cell adhesion molecules occur, which are basically cadherins, integrins, selectins, and the immunoglobulin Ig superfamily. The adhesion of cell to cell may be calcium dependent, which are seen in cadherins and selectin, whereas in some cases it is independent. For example, interaction involving integrins and immunoglobulin superfamily CAMs do not require calcium ions. Now, coming to cadherin. Cadherin are calcium dependent cell adhesion molecules, and these are involved majorly in cell to cell adhesion, signaling, and tissue differentiation. Removal of calcium ions leads to dissociation of cadherins attachment. This family of protein contains more than 100 members among vertebrates and invertebrates. Variety of cadherin molecules is possible because of the presence of multiple genes as well as alternate RNA splicing that results in number of mRNA from single gene. In general, cadherin is a type 1 integral membrane glycoprotein of approximately 720 to 750 amino acids. Among all members, cadherin family E, cadherin P and N cadherin are classical cadherins and most widely expressed. E cadherin is abundant among lateral surface where they connect adjacent cells. Now, coming to integrins. Integrins are the adhesive molecules which allow cells to maintain contact with one another and with structure in the extracellular matrix. These are basically diverse family of transmembrane proteins found in all animal cells and are involved in cell extracellular matrix interactions through focal adhesion and hemidesmosomes. I will talk about the focal adhesion and hemidesmosomes after some time. Each integrins molecule is composed of two main glycoprotein parts, a alpha subunit and a beta subunit, that are associated through non-covalent interactions. Variation in the alpha and beta subunits accounts for the wide variety of integrins observed throughout the animal kingdom. For example, a diverse range of human integrin heterodimers are found as a result of combination of 9 beta subunits and 24 alpha subunits. Multiple integrins are required for recognition of many matrix proteins. For example, at least 8 integrins bind to fibronectin and 5 to laminin. Cell-specific integrins receptors may interact with other factors that simultaneously change their specificity for same ligand. As attachment of cell require attachment with cytoskeletal protein and with the matrix protein, integrins act as a receptor for matrix component. Integrins link the actin cytoskeleton of cell to various external structures. If you see in the figure, you will find that the cytoplasmic portion of each integrin molecule binds to adaptor protein that connect to the actin filamen inside the cell. The extracellular portion of the integrin then binds to molecules in the extracellular matrix or on the surface of other proteins. Integrins attachment to neighboring cells can break and reforms as a cell moves. And this figure shows the integrins connects with the extracellular matrix with actin cytoskeleton inside the cell. Now, other types of cell adhesion molecules are selectins, which are lectin containing glycoproteins that are involved in cell to cell adhesion in bloodstream. Lectin domain of these transmembrane protein is conserved and specific for binding with surface oligosaccharides of other cells. There are 3 types of selectins, each is specific for the type of blood cells. For example, L-lectin is selective for WBCs, P for blood platelets and endothelial cells, which is activated by inflammation, and E-selectin, which is specific for endothelial cells. L-selectins are the smallest and expressed on most of leukocytes. P-selectins are the largest selectin molecules and stored in the granules of platelets and Weber pallid bodies of endothelial cells. P-selectins are translocated on the cell surface of activated endothelial cells and platelets, while E-selectins are generally not expressed under normal conditions except for in skin macrovessels, but it is induced by inflammatory signals. Functions of selectins involve crawling movement of WBCs for constitutive homing. This is shown in this figure where the P-selectin expressed on endothelial cells act as a receptors for P-selectin's glycoprotein ligand 1 on the surface of leukocytes. Binding of P-selectins with the PSGL1 allow WBCs to move over the endothelium cells. Selectin expressed on surface of endothelium cells are required for the movement of WBCs, and this figure clearly shows this pattern. The last family of cell adhesion molecules is immunoglobulin superfamily, or that's we call the IDSF, which is involves in the large family of surface and soluble proteins. Members of this superfamily have common structural features similar to immunoglobulins and include receptors for antigen, co-stimulatory molecules, cell adhesion molecules, cytokine receptors, and molecules of antigen presentation. Cell adhesion molecules of this superfamily plays critical role in cell adhesion and are also associated with cancer. This family of immunoglobulins superfamily includes intracellular cell adhesion molecules, number 2 vascular cell adhesion molecules, number 3 nerve cell adhesion molecules, number 4 synaptic cell adhesion molecules, and platelet endothelial cell adhesion molecules, as well as CD2 and CD48. ICAM1 is present on the surface of leukocytes and endothelial cells in low concentration. Cytokines like interleukin 1 and tumor necrosis factor act as a stimulatory signals for expression of this ICAM1 on the surface of vascular endothelium and macrophages. ICAM1 is an important for stabilizing cell to cell interactions, and it facilitates leukocyte endothelial transmigration. While vesicle VCAM1 is required for attachment of eosinophils, basophils, monocytes and lymphocytes to the vascular endothelials. Their expression also increased by cytokines like interleukin 1 and tumor necrosis factors, while neural or nerve NCAM are required for neuron-neuron or neuron-muscle adhesions. These molecules are important during development, including cell proliferation, differentiation, and migration. If you see that, like Cadherins, NCAM primarily mediates homophilic interactions, which shows the binding together cells that express similar NCAM molecules. But unlike Cadherins, NCAM are encoded by a single gene. Their diversity is generated by alternate mRNA splicing and by differentiation or differences in glycosylations. Now coming to the cellular junction with the second part, that there are different junctions which are present in different cells, which forms the networks to the nearby cells, and they contain a different protein makeup. The adhesive transmembrane proteins anchoring these junctions have extracellular portion that interact with similar proteins on adjacent cells. Protein complexes within each cell further connect the transmembrane adhesive proteins to the cytoskeleton. There are different types of cell junctions are present, which either link cell to cell together or linking cell to the basal lamina, and this is showing in this figure. There are different types of cell junctions which are shown either linking individual cells together or linking cells to the basal lamina. A view inside of cuboidal cell in a corner is shown in the center of the diagram together with a cell of each side of it. All three of the cells are sitting on basal lamina, which is shown in dark pink color. Tight junctions, adherens junctions, desmosomes and gap junctions are shown bridging the gap between the plasma membrane of the central cell and the plasma membranes of its two nearby cells. Hemidesmosomes are shown between the plasma membrane of each of the three cells and the basal lamina below them. Tight junctions are depicted as a small blue sphere within the plasma membrane of adjacent cell. This figure shows 3 points of contact between two cells at the tight junction, and at each point of contact a blue sphere from one cell is connected to the sphere from the adjacent cell. The tight junctions lie on the side of the cells closest to the cell apical surface. Adherence junctions are shown below the tight junctions. The adherence junctions are depicted as horizontal blue lines that lie across the plasma membrane. 5 blue lines from each of 2 adjacent cells integrate between the 2 cells, linking the cells together. The adherence junctions are linked to a band of actin and myosin filament, which are shown as horizontal red lines running parallel to one to each other along with the inside of the cell membrane. Desmosomes are shown in green and lie below the adherence junction. The desmosomes of adjacent cells are shown as globular structure within the interior of the cells with horizontal lines extended across the membrane and shows as a space between the cells. Short green lines are connected to the intracellular portion of desmosomes and they represent the intermediate filaments. The globular desmosomes together with the extended intermediate filaments arms appear like spider-like. If you see in the figure that the gap junctions are also shown in the membrane of adjacent cells. The gap junctions are shown in yellow and consist of multiple channels that are made up of 6 round of subunits that surround a central circle. The gap junction channels are shown from the side as a pair of white lines across the plasma membrane of adjacent cells. Hemidesmosomes are also shown on the basal surface of these 3 lines, or basically 3 cells. They are shown as blue globular structure with the vertical blue lines crossing the plasma membrane and containing the basal lamina below the cells. Short blue lines connecting to the intracellular portion of the hemidesmosomes represent intermediate filaments. So this can show the comprehensive structures of all junctions which are generally present inside the cell or towards the basal lamina or near to the adjacent cells. So now we'll come one by one to different junctions. Number 1 is the anchoring junctions. These junctions contain surface glycoprotein that link with the cytoskeleton through adapter proteins. These junctions are involved in cell-to-cell adhesion and cell-to-matrix adhesion, as we have seen in the previous figure. These junctions also provide mechanical strength to the cell structures. Based on the presence of specific cytoskeleton filament, anchoring junctions can be segregated into 2 types. Number 1, adherence junctions, which may contain the epithelial cells and a continuous band of cadherin molecules, usually located near the apical surface just below the tight junctions that connects the lateral membrane of epithelial cells, and this junction is known as adherence junction. Classical cadherin molecules contain single transmembrane domain with short C-terminal cytosolic domain and 5 extracellular domain. You can see this thing in figure where the organization of surface cadherins and the 5 molecules are attached to each other. Now, this extracellular domain is capable of binding with calcium ions and required for cell-to-cell adhesion. Each extracellular domain is composed of 5 to 6 repeats of cadherin proteins, and calcium ions place inside each pair of cadherin to form a stiff rod-like structure. The C-terminal cytosolic domain is linked to actin filament of cytoskeleton through the intracellular anchoring proteins like alpha, beta or gamma catenin. These interactions are important for strong cell adhesion, as disruption of adapter proteins will dramatically decrease cadherin mediated cell to cell adhesion. The adapter beta catenin not only act as an adapter protein for attachment with cytoskeleton, but also important molecule of signaling that enters inside the nucleus and later gene expression. Intracellular networks of all interacting proteins, cadherin of both cells interact directly during a cell to cell adhesion through adhesion junctions. A deletion mutation of p-cadherin encoding a gene, that's CDH3, leads to hypotrichosis with juvenile muscular dystrophy. This autosomal recessive disorder initiates with early hair loss and followed by retinal muscular degeneration that leads to the blindness after the age of 20 years. Now we have another junction that we call a desmosomes. A desmosome consists of proteinous adhesion plaque, which is 15 to 30 nanometer thick, attached to a cytosolic phase of the plasma membrane of adjacent cell and connected by a transmembrane linker protein. Plakoglobin and plakophilin are the major constituents of these plaques, which are similar to beta catenin, and they act as anchoring proteins. These anchoring proteins bind to specific cytosolic cadherin proteins, which are desmosomes and desmocholine, and extend into the intracellular space, where they interact, forming an interlocking network that binds to two cells together. These cadherins are differed from classical cadherin as they have unrelated in their sequence throughout the intracellular as well as the extracellular domain. Intracellular network of all interacting protein, cadherin of both cells interact directly during the cell to cell adhesion through the desmosomes. Importance of desmosomes and related cell adhesion molecules can be understood in patients which are suffering from pemphigus and autoimmune disease, where the autoantibodies against desmosomal cadherin proteins form. Affected patients have severe skin blistering along with the fluid secretion from loosened epithelial. This figure shows a patient of such disease, pemphigus, where there is a problem with the cell adhesion proteins. Now, we have another type of cellular junction that we call as focal adhesion and hemidesmosomes, which were earlier seen in the figure. Focal adhesions are subcellular integrin containing macromolecules assemblies through which signals are transmitted from extracellular matrix to a cell. They act as a mechanical linkage to the matrix, biochemical signaling hub that direct several signaling proteins at the position of integrin binding. State of constant flux is maintained at focal adhesions, where protein associate and dissociate simultaneously. Upon signal transmission, about 100 different proteins are present in a focal adhesion and hence accountable for their functional diversity. Interaction of integrins with proteins of extracellular matrix involve binding with a specific sequence, for example, RGD sequence, which is known as arginine, glycine, aspartic acid motifs of fibronectin, or DGEA and a motif of collagens. Binding of integrin with receptor recruits anchoring proteins along with focal adhesion kinases. These anchoring proteins include talin, tensin and vinculin that further interact with actin filament during signaling cascade to regulate signal functions. This figure shows the interaction network of cellular proteins upon binding of integrin receptors to their extracellular ligands. Now coming to another molecule that we have hemidesmosomes. These are very small structures and found in keratinocytes of the skin epidermis that are in association with extracellular matrix. Structure assemblies of hemidesmosomes is similar to focal adhesions. The only difference is in the type of cytoskeleton filament and anchoring protein. Hemidesmosomes are attached with intermediate filament instead of actin filaments and plankton, and dystonin are intracellular anchor proteins in hemidesmosomes. The present table shows are different types of cell adhesion molecules. They are related anchoring proteins, they are related junctions and their type of interactions, and lastly the filaments which are being utilized in this one. Number 1, if you see the cadherins, they may be classical cadherins or they may be desmoglin or desmocholine. In classical cadherin, alpha, beta and gamma catenins are required as anchoring proteins and they are required for adherence junctions for cell to cell adhesion, and their related cytoskeleton filaments are actin filaments, while in case of cadherins which are desmoglin or desmocholine, they require a plakoglobin and plakophilins anchoring proteins. They form in desmosomes and require for cell to cell adhesions, and for the molecules of cytoskeleton filaments required, they have intermediate filaments. If you talk about integrins, which are also cell adhesion molecules, they are of different types, and the anchoring proteins for these are talins, tensins and vinculin. They are required for focal adhesions. Their interactions are cell to matrix adhesions, and the related cytoskeletal filaments are actin filaments. And if you talk about alpha 6 beta 4 cell adhesion molecule, they are required for plankton and dystonin anchoring proteins which are present in hemidesmosomes and required for cell matrix adhesion, and for these molecules intermediate filaments are required as a cytoskeleton filament. Another junction that we call occluding junction or tight junctions. If you see that if cells are required to exchange materials between them simultaneously, then why tight junctions are required? The answer lies for this matter is internal cavity of gut contains specialized absorptive cells that are capable of absorbing nutrients from lumen cavity. Then absorptive cells are required to transport nutrients into extracellular matrix so that they can diffuse into blood vessels. This transcellular transport requires two sets of transport proteins, one at apical surface for active transport from gut lumen, and second at the basolateral surface for facilitate diffusion of same molecule to extracellular matrix. Effective transport is only possible if adjacent epithelial cells are tightly packed so that transported molecules cannot leak out back into the lumen. This can be easily achieved with the presence of occluding junctions, or which are also known as tight junctions, which were shown in previous figure as well as in the coming figure. These junctions are found in vertebrates. Those are required for sealing of two cells that act as barrier for passage of any molecules. Same function is performed by separate junctions in invertebrate. Branch network of independent sealing strands are required for the formation of these junctions. Hence, sealing ability to resist ion passes eventually increases with the number of strands. Each strand is formed from a row of transmembrane proteins that are embedded in cell membrane of both cells. This figure shows a comprehensive structure of occluding junctions or tight junctions, which shows the sealing cells together. Transmembrane proteins that act as a building blocks for formation of these sealing strands are claudins, occludins, and tricellulin. The claudin protein family has 24 members in human that are expressed in combination with different epithelia to confer permeability to the epithelia. A dominant mutation in occludin may disrupt structures of tight junctions and hence result in associated diseases. Crohn's diseases, which is an inflammatory bowel disease, is a result of disruption of intestinal mucosal barrier that is maintained by tight junctions. Lastly, we will talk about a junction that we call the gap junction. For the continuous exchange of ions and biomolecules between neighboring cells, it takes place through gap junctions that forms 2 to 4 nanometer channel between the membranes of adjacent cells. Presence of gap junctions maintained direct connection between cytoplasm of interacting cells that is required for transport of molecule, which is essential for cell communication and maintenance of homeostasis. Gap junctions are present in vertebrates as well as invertebrates except for plants, where Plasmodesmata carried out similar function. Structural assemblies of these junctions in chordates and non-chordates is not similar. For example, annexins are present in non-chordates as a substitute of connexins, which are present in chordates. Although structure of gap junctions are different, however, channel form from annexins and connexins are similar. If you see the organization of these gap junctions, you will find that 4 transmembrane domains of connexins are required to form a pore of a channel. These domains further connected with the extracellular loop. Extracellular loops are required for cell recognition. Assembly of 6 connexin molecule form a hexagonal structure on a cell surface called connexon or hemichannel. This is shown in this figure, where the joining of 2 connexons from different cell surface will form a channel of gap junction, which results the formation of a pore like structure between the 2 adjacent cells. Now, you will find that there are about 21 isoforms of connexin genes which are present in human genome and are required for expression of different connexins on different cell types. Expression of different genes result in the different homotypic or combination of heterotypic gap junctions, and it forms the diversity of gap junctions, which results in the permeability differences. For example, CX43 isoforms result in a 43 chloraldehyde connexin that is 100-fold permeable to ATP and ADP as compared to 32 kDa connexin, which are expressed from CX32. Importance of gap junctions can be visualized as a look after patients with mutations in connexins that result in various skin abnormalities. For example, keratitis Ichthyosis deafness syndrome is an autosomal dominant disorder associated mutation in connexin 2. Symptoms for this disease include red, rough, thick plaque on skin and neurosensory deafness that further leads to photophobia. Now, let us summarize what we have learned from this module. Number 1, adhesion of like cell is a primary feature of architects of many tissues. Number 2, interactions of different two cells require multiple ligands and adhesion molecules, which are diverse group of integral membrane proteins, which are known as cell adhesion molecules or CAMs. Cell adhesion molecule are of different classes like cadherins, integrins, selectins, and immunoglobulin superfamilies. Cadherin and selectins are responsible for calcium ion dependent homophilic interaction between cells, while calcium independent homophilic interactions between cells are mediated by nerve cell adhesion molecules, which are belong to the family of immunoglobulin superfamily, and the similar things will be seen in integrins. Selective binding. Selectin binds to the carbohydrate group on mucin like cell adhesion molecules, while their distal lectin domains, which mediates calcium dependent heterophilic cell to cell interaction. In all cell adhesion junctions, clusters of transmembrane cell adhesion molecules are linked via various adapter molecules in cytoplasmic plaques to the cytoskeleton. Adherens junction and desmosomes are cadherin containing junctions that bind membrane of adjacent cells in a way that gives a strength and rigidity to entire tissue. Continuous exchange of ions and biomolecules between neighboring cells takes place through cap junctions that form 2 to 4 nanometer channel between membranes of adjacent cells. Cap junctions are constructed of 12 copies of a single protein that we call the connexins, which is formed into a transmembrane channel that interconnect the cytoplasm of two adjacent cells. Presence of gap junctions maintain direct connection between cytoplasm of interacting cells that is required for transport of molecules, which is essential for cell to cell communication and maintaining homeostasis. Thank you.